to the fisheries, fish breeding, is it not? So we have learned this common carp, then grass carp, silver carp. All these ones were reared in Indian waters, in the sense either sea or fresh waters. And this breeding of fishes has to be done for more amount of fish. So this rearing of fish can be done by hypophysation or induced breeding. That is for artificial breeding. Okay. So what these people have to do in this hypophysation or induced breeding, in the sense, actually a special arrangement will be made for this artificial breeding. These fishes are actually acclimatized to release their eggs and sperms in running waters. So what has to be done, in the sense, while they are going for artificial breeding, the matured ones, matured brood fish, both male and female, will be selected. And then artificially running water will be created and they will be let in to that water. Just like waterfalls, artificial waterfalls will be created in brooding center. And these brood fish, both male and female, will be left out in that artificial waterfall area. Why? Because these ones are acclimatized to release their sperms and ova in running water. So prior to the release of these brood fish into the water, pituitary extracts containing gonadotropins, such as follicle stimulating hormone and luteinizing hormone, that is in short form FSH and LH. These ones will be injected into these brood fish. Why? Because whenever we want to have sperms and ova from these brood fish, it will not be possible. For that condition, what we are supposed to do, in the sense, we have to inject these types of hormones such as FSH and LH. These ones will be extracted from pituitary. Okay, or even ovaprim also can be injected. This ovaprim is nothing but it is a synthetic analog of these hormones, almost equivalent to these FSH and LH, but artificially prepared hormone that is known as ovaprim. Okay, so this either FSH or LH, FSH and LH, or artificial ovaprim have to be injected into the both male and female fish, brood fish, and they will be released into the running water. And then these fishes will release their eggs and sperms, and in the running water itself, fertilization will occur, and the zygotes will be the resultant. And from those zygotes, fingerlings will come out. A small fish, just like small fish, will be coming out, and those ones will be transferred to another tank, such that they can be reared artificially. Okay, so that is how the induced breeding in artificial breeding will be done. So what we are supposed to do, in the sense, we have to inject gonadotropins. Then only these brood fish will be able to release sperms and eggs. Okay? And now we will go for economic importance of fisheries. As you know, these pies is nothing but fishes will be eaten as food material, fish meat, So this fish meat in general, it is a very good source of proteins, which can be digested very easily, and vitamins like A and D, minerals, and rich in iodine. Okay? So these are all seen in fish meat. Hence, yesterday I told you, doctors will be prescribing to their general persons just to go for fish meat instead of going for mutton and chicken. Okay? Why? Because this protein, which is available in fish, can be digested very easily. And added to those ones, vitamin A and D also will be available, minerals, and rich in iodine. So this iodine is useful for thyroid gland. Okay? And added to these food products, byproducts such as shark and cod liver oil will be containing vitamin A and D. So this is a great thing. So shark liver oil and cod liver oil are good sources of vitamin A and D. Then not only that, oil from sardine and salmon fish are good sources of omega-3 fatty acids. You nowadays people are having health sense, and these people are suggested to go for omega-3 fatty acids, which are helpful for human health, which have multiple functions. So even people will be taking or gulping capsules which are containing omega-3 fatty acids. Instead of taking tablets, you can take these sardine fish and salmon fish, which are good sources of omega-3 fatty acids. And the other one is fish guano. Here, fish guano is nothing but a scrap of a fish, and it is prepared as fertilizer. That will be ultimately known as fish guano, scrap fish. Okay. And next one is shagreen and isinglass are other byproducts. What are these shagreen and isinglass in the sense? Here, shagreen is the dried skin of shark. The name itself will be denoting that it is dried skin of shark. And isinglass is the substance that is obtained from dried swim bladders of catfish. Generally, these catfish will be containing swim bladders such that they can float on the surface of the water whenever they want. So naturally, these catfish will be containing the swim bladders. So from these swim bladders, the substance will be obtained and it will be dried. That will be ultimately known as isinglass. So both of these shark material and catfish material, nothing but shagreen and isinglass, are the subproducts of Pisces. And then these ones are used for clarification of wines. What is meant by clarification of wines in the sense? While preparing wines, suspended particles will be there, which will be appearing or will be making the wine to be not clear, but gloomy. Imagine a water bottle having all muddy particles or suspended particles. How it will look? It will look odd, no? So in the same way, even in wine bottle also, suspended particles will be remain. So just to take out those suspended particles in wine bottles, this shagreen and isinglass will be used. And those ones are extracted from shark skin and this catfish dried swim bladders. Okay, I hope you got the point. So these are the uses of this fish. Okay, And nowadays, here, as you know, the rearing of fishes, brooding, then artificial brooding, everything will come under blue revolution. So you have heard about green revolution. Who is the father of green revolution? MS Swaminathan. So that here, just because of green revolution, we are able to have more amounts of crop products, is it not? In the same way, rearing of fish to the maximum extent can come under or can be kept under blue revolution. So this blue revolution nowadays is implemented on lines similar to green revolution. How far we are going for green revolution, in the same way we are going for blue revolution. Why? Because most of the people are of non-vegetarians. So particularly in this non-vegetarian food also, fish meat is very good and healthy food material. That's why nowadays people are going for fish meat only. Hence, it is the responsibility of the country to go for blue revolution. And here, Hiralal Chowdhury and Arun Krishnan are taken as fathers of blue revolution. How MS Swaminathan is considered as father of green revolution. In the same way, Hiralal Chowdhury and Arun Krishnan are termed to be the fathers of forefathers of blue revolution. Okay, so this is about the fisheries. Okay, till now we have learnt about fisheries, then poultry, then beekeeping, then livestock. All these ones we have learned. Now we'll go for biotechnological applications in medicine. How we are going to utilize biology plus technology, both of these ones, for our health purpose. Till now we have seen only for our food purpose. Now we are going for applying biology and technology in medicine, in the sense for our health purpose. Okay, what are all the things? Why we are supposed to go for medicine, in the sense, in what circumstances we are supposed to go for medicine, in the sense, when we are sick only, is it not? So when a person becomes sick, he has to take medicine. And what are all the medicines that have to be utilized for curing that sickness? All these ones, in all those aspects, we are supposed to utilize this biology, knowledge of biology and technology. Both side by side, they have to be utilized for applications in medicine also. Okay, so for this purpose, primarily recombinant DNA technology is utilized. So this recombinant DNA, in short form rDNA technology, is involving manipulation of genes or microorganisms to produce certain products useful for mankind. Here, priorly itself, I told you, gene is responsible for characters. So these characters of human beings' body can be manipulated by involving rDNA technology. So that is the process seen here. Okay? So rDNA technology involves manipulation of genes, or genes of human beings, or even microorganisms also we can manipulate to produce certain products useful for mankind. These ones are used in production of hormones, synthetic vaccines, effective therapeutic products, etc. Okay, so these biotechnological applications will be useful in production of hormones. So here how it will be done is shown in this picture. So insecticidal cells have to be selected. Cells have to be selected, and they are injected with the material that is here. We will be using recombinant DNA technology, and then we'll be manipulating the genes that are seen in those normal cells, and ultimately they will be converted into insecticidal cells. And those insecticidal cells will be incorporated into the plants which we are going to rear. And these ones, if they are fed with insects which are causing harm to those particular plants, naturally they'll die. Why? Because priorly itself we have selected insecticides. So these insecticides have been incorporated into normal plant cells which we are going to grow, and those ones are injected into the plant body. And whenever the plant body is, or either leaves or stem or something like that, is eaten by the insects, which will be causing harm to our crops, naturally by eating those ones, they will die. So in the same way, here you might have heard about BT cotton. So this BT cotton, why we are growing, in the sense, stem borer will be there, pink bollworm, and just to kill those pink bollworm, we need not spray any insecticide or pesticide or something like that. Just by using rDNA technology, we can make these insects to die. In the same manner, even for human beings, medicines also can be utilized. Okay. So the medicine or the material which has to be utilized by human being also can be done by rDNA technology. For example, you can go for insulin. In lower classes itself, you came across this insulin. Here, it is considered as a protein hormone. So this protein hormone, which is termed to be insulin, is produced from beta cells of islets of Langerhans of pancreas. And this insulin is must for regulating the glucose content in our body. So if it is not there, insulin is not there, naturally people will get will land up as diabetic. Our Indian population, most of the Indian population is having the disease, that is, it became common. Having diabetes is becoming common nowadays in Indian population. Why? Because this insulin is not produced from beta cells of islets of Langerhans of pancreas. Okay. So what we are supposed to do, in the sense, we have to inject insulin, but we cannot take insulin oral. Why? Because it is a protein hormone. So if protein hormone is taken as tablet, it will reach our stomach, then digestive tract, and it will be digested before it gets absorbed. Hence, we cannot take this insulin in the form of tablet. What we are supposed to do? Now we are supposed to inject the insulin in the injection form. Okay. So here, this insulin, in olden days, they used to collect from cattle, pigs. That means livestock. So just to collect the insulin from livestock, cattle and pigs, we have to slaughter those ones. It will be a great loss to the mankind, is it not? So they can be utilized in other forms, but only for insulin purpose, if you are slaughtering these higher animals, it will be a great loss. Other uses cannot be taken from these animals. That's why scientists have experimented, and they have found out the responsible gene which is responsible for secreting insulin hormone, and they have taken out that gene, and they have injected this gene, genetic material, by rDNA technology into a bacterium that is known as Escherichia coli. So this Escherichia coli can be reared very easily in a great number. Okay, so what we are supposed to do in the sense here, we have to utilize this rDNA technology, and then we have to incorporate that insulin producing gene into the plasmid of Escherichia coli. Okay, then ultimately it will be giving us the insulin. Okay, so that is the technology utilized here. If we want to go for the structure of insulin, it will be containing, as I told you already, it is a protein hormone. Naturally, you can expect amino acids. Why? Because amino acids are nothing but the minute particles of proteins. So here 51 amino acids will be there, and which will be arranged in 2 polypeptide chains. That is chain A and chain B. Chain A, chain in the sense first chain will be containing 21 amino acids, whereas the second chain, that is chain B, will be comprising of 30 polypeptide chains. Okay, amino acids. 30 amino acids in chain B and 21 amino acids in chain A. These two are attached with polypeptide chains. And added to these ones, here when we are going to combine these 2 polypeptide chains, which contains an extra stretch called C peptide also will be seen. So this C peptide will be seen in proinsulin. Proinsulin in the sense, the prior stage for proper insulin. Insulin that is utilized or prepared in human being can be termed as humulin. So prior to this humulin formation, one more chain will be there that is ultimately known as C peptide chain. Okay. Now A peptide chain is there, B peptide chain is there, and the C peptide chain also will be there. So this C peptide chain can be seen only in pro-insulin condition, but it will not be seen in proper insulin condition. Here, what's the problem in the sense? Combining these two, that is chain A and chain B, is the problem. For that purpose, what they have done in the sense, here these 2 polypeptide chains have been introduced into Escherichia coli by a great American company that is known as Lilly. So this company has succeeded in combining these 2 chains by incorporating these ones into the plasmids of Escherichia coli, and they have successfully done it. And the resultant is creating disulfide bond to form humulin. As I told you, humulin is nothing but the human insulin, is technically known as humulin. So this company people have succeeded in combining these disulfide bonds. In the sense, combining these A chain and B chain, having 21 and 30 amino acids, and forming this humulin. So this is how this biotechnology can be applied in our medicine. Okay, so this is a simple example I told you already. Okay, and now we will go for vaccines. As I told you here, it will be used in preparation of hormones. We have seen the example of insulin. Now we'll go for vaccines. Okay, so how it can be done? First of all, what is meant by vaccine? A vaccine is a biological preparation that improves immunity to a particular disease. Okay, so prior to the getting of disease itself, we'll be going for vaccine. Okay, so prior to the getting of disease itself, we have to take vaccine. That is, in one way or rough way, we can tell that vaccine is a precautionary medicine just to avoid the disease which is going to get into our body. Okay, so a vaccine is typically containing disease-causing microorganisms, often made weakened or killed forms of microbes. So as you know, diseases will be caused by microorganisms such as virus, bacteria, fungus, everything. Okay, so these ones will be made weak or they can be killed. And weakened ones or killed ones of these microbes which are going to cause diseases to us will be injected as vaccine. See the nature here. Okay, so another way of this one is the toxins are one of the surface proteins of microorganisms are also used in preparing vaccines. I told you already here, weak ones or killed ones of these microbes which are going to cause disease will be accepted as vaccine. Not only that, the toxins are one of the surface proteins of microorganisms are also used in preparing vaccines. That means from these microorganisms, the skin, rather to say surface of, suppose imagine a virus which will be having protein pokes. Nowadays, we are getting worried about this COVID. Is it not? So pokes of these virus surface proteins can also be utilized in preparing vaccines. Okay. And here vaccines are generally used as prophylactic measures. As I told you, these are preventive medicines. And coming to this usage of vaccine, use of vaccines has reduced mortality rate to a great extent. Here in Asian continent, many of the people used to die just because of smallpox priorly. Okay, so due to this smallpox, many people died in our continent, Asian continent itself, not only India, Asian continent. So this vaccine against this smallpox has eradicated the disease itself to the greater extent, and mortality rate has been reduced to the greater extent. Okay, so that is there. So here, for example, smallpox could be eradicated completely by intense vaccination program in an organized manner, as in the case of polio. Since we are not having smallpox, we are not worried about this one. But nowadays, polio vaccines are given to the young child, just born child, to more or less 5 years old child. And every month, the government will take up a particular day and they will go for vaccination of polio. And this will be given out as advertisement by great people such as Amitabh Bachchan or so. Okay, so polio vaccination, just 2 drops of polio vaccine will protect the child to avoid this dreadful disease that is known as polio. So like that, here the vaccines are utilized for human beings' welfare. And here, though there are vaccines available against several pathogens, vaccines against diseases such as AIDS and malaria are yet to be developed. Why? Because just we have reduced the mortality rate in the case of smallpox, and almost we have succeeded against polio. Polio percentage is reduced to the great extent. But nowadays, so many people are dying just because of AIDS, acquired immunodeficiency syndrome, and even malaria also. That is spread by this Anopheles mosquito, is it not? So just due to these 2 diseases, many people are dying nowadays. So we are supposed to bring out the vaccines against diseases of AIDS and malaria. They are yet to be developed. Okay. And here some of the important biotechnologically produced vaccines are there. These ones are 3 types. The first one is attenuated whole agent vaccine. How they are prepared, based on that only these ones are divided. The first one is attenuated whole agent vaccine. This will be containing disabled live organisms. Okay, so disabled live organisms. For example, vaccines like MMR, typhoid, yellow fever. These 3 are termed to be attenuated whole agent vaccines. MMR in the sense measles, mumps, and rubella. So these 3 diseases can be avoided by taking a MMR vaccine. And of course, typhoid, you know very well. Yellow fever also, you know. So these diseases, vaccines that avoid these 3 varieties of diseases will be containing disabled live pathogens. Okay, so that is termed as attenuated whole agent vaccines. And coming to the second variety, inactivated whole agent vaccine. Actually, they contain killed microbes. The microbes which are causing cholera, polio, rabies, hepatitis A, all these ones, vaccines, will be containing killed microbes. Okay, so killed in the sense the cholera, for example, if you take up Vibrio cholera is the bacterium which will be causing this cholera. That microbe will be killed and will be injected into our body such that we won't get cholera. Okay, so that type of vaccine is termed to be inactivated whole agent vaccine. Hope you got the point. And the third variety is the toxoids. So they contain inactivated exotoxins of certain microbes. Just prior to this, I told you already, here these vaccines are generally used as toxins of surface proteins, I told you, of microorganisms. So surface proteins of these microorganisms will be taken, and then those ones are utilized as vaccines. That is nothing but inactivated exotoxins of certain microbes. So exotoxins, exo outside. So microbes from outside of the microbe's body, the substances will be made, and those ones will be inactivated, and they will be given as vaccines. For example, TT. TT is nothing but tetanus vaccine. Okay, tetvac is TT. And added to that, diphtheria also will be taken as exotoxin of the microbes. Okay, that comes under toxoids. So this is about the vaccines preparation. 3 varieties are there: attenuated, inactivated, and toxoids. Okay, so that is the thing. And now we'll go for molecular diagnosis. If we want to have a clear, effective treatment of a disease, diagnosis has to be done. You might have heard this term diagnosis, where near doctors, in the sense doctors have to diagnose the disease and then they have to give medicine to us. That means what type of disease is there, or what type of disease causing organism is residing in our body and causing disease has to be found out by doctors. So that is actually diagnosis. So if the doctor is not able to diagnose the disease seen in our body, he will give some other medicine, not correct medicine. Is it not? Then we won't get rid of the disease, particular disease. Okay. So here, what has to be, or what is the important thing in this nature is diagnosis has to be done clearly. And even that diagnosis has to be done at early stage. If it is late stage means, then no use. By the time we find out the disease causing organism and take medicine, that medicine will not be useful. Why? Because our full body will be filled with the disease causing organisms. Is it not? That's why early diagnosis is very important. For this also, we can utilize this biology. Okay. So using conventional methods of diagnosis, it will not be possible for early detection of the disease. Actually, here what we will do, we'll go for blood test, urine test, all these ones. Okay. To some extent, it will be utilized, but not always. So early detection, if it is done, medicines can be given and the disease can be cured. But if it is not detected early, then the whole body will be filled with, in the sense, our body will be taken over by the disease causing organism. It will go into the hands of the disease causing organisms. But conventional methods are nothing but blood test and urine test. So nowadays, these tests are of not to the greater use, but only less. Primary utilization will be done. Added to these serum, in a sense, blood and urine tests, we have to go for rDNA technology, PCR test, that is nothing but polymerase chain reactions, are some of the techniques which help in early diagnosis. Okay. So just in olden days, we used to go for blood test and urine test. Of course, nowadays also we are going for. But if we are unable to detect out the disease, then naturally we have to go for recombinant DNA technology and PCR test, that is polymerase chain reaction test. Okay. So these are the techniques which will be utilized in early diagnosis. Okay. And presence of pathogen is normally suspected when the pathogen produce disease symptoms. Generally, how will find out that we got disease in the sense, particular disease will be having particular symptom, is it not? So just by seeing that disease symptom only, we'll be able to come to know that we got some other disease. Okay. So when it is done, when pathogen enters into our body and it lives as it is in our body only, that disease symptoms will be shown outward. So by this time, the concentration of pathogen is already high in the body. So by the time we wake up and then we find out, we observe our disease symptoms, by that time, the number of pathogens or concentration of pathogens will be very high. If it is the thing, then we can do nothing. Is it not? So, however, very low concentration of bacteria or viruses can be detected by amplification of their nucleic acid by PCR test, that is polymerase chain reaction. So here, as soon as we get disease symptoms, it is better to go for PCR test. Why? Because very less amount of concentration of bacteria or virus also can be detected out by PCR test. Okay, this PCR helps to detect out very low amounts of DNA by amplification of small DNA fragment also. So just by taking of little amount of tissue itself, we can find out, in the sense tissue, in the sense blood also. So this blood is taken out, and then we are going for PCR test. We can find out the pathogen which is residing in our body. Okay. You might have heard this PCR term in the case of HIV patients. So just to detect out HIV, that is human immunovirus, PCR test is nowadays done. Okay. So in suspected cases, we'll be going for PCR test. And this one is used to detect out mutations in suspected cancer patients also. So even in cancer patients also, just to detect out mutations which are going to occur also can be detected out by PCR test. And it is a powerful technique to identify many other genetic disorders such as hemophilia, phenylketonuria. So these varieties of diseases can be detected out by PCR test. That is a very powerful technique just to identify even genetic disorders also. Here we are talking about HIV, that is nothing but a viral one. And the previous one is actually here some of the diseases such as the MMR, in the sense typhoid or yellow fever or rabies, polio, all these ones, just to find out those varieties of diseases, we will be utilizing this PCR. Why? Because here the causative organism will be there. Whereas in the case of genetic disorders, causative organisms will not be there. Our genes itself, they will be malfunctioning and they will be causing diseases to us, such as hemophilia, where the blood will not be getting clotted. So this type of genetic disorders, just to find out those disorders also, we are supposed to use PCR test. That is nothing but polymerase chain reaction test. And here, how it will be done, in the sense, a single-stranded DNA or RNA tagged with the radioactive molecule, and it will be allowed to hybridize with its complementary DNA in a clone, and it is followed by its detection using autoradiography. Okay, so this autoradiography is the technique which will be utilized in the detection of the disease. Okay, and the clone having mutated gene will not appear on the photographic film because the probe does not show complementary with the mutated gene. So by using this PCR technique, we will be able to find out the cause of the disease. Okay, so that is the mechanism. So that is the thing. And now we will go for gene therapy. So this gene therapy is nothing but insertion of genes into individual cells or tissues to treat disease such as hereditary disease. So for example, if you take up the hereditary disease, that means here what are all the diseases that will be just getting hereditary, in the sense generation after generation, every generation, if it is getting the particular disease, then it can be called as hereditary disease. So how these hereditary diseases can be treated? Just by gene therapy. In the sense, simply telling, we have to detect out the gene which is responsible for that particular disease, and we have to take out that particular gene and incorporate a healthy gene in that vacant place. So that is the simple technology which will be done in gene therapy. Okay, so gene therapy is the insertion of genes into the individual cells or tissues just to treat the diseases which are of hereditary in nature. So generation after generation, if the disease is getting continued, then it can be called as hereditary disease. And these diseases can be treated by a particular mechanism that is known as gene therapy. What it will be done in the sense, the disease causing gene will be detected out and it will be taken out, and in that vacant place, a fresh, healthy gene of the same type will be inserted. That is nothing but gene therapy. Okay. So the biology of human gene therapy is a collection of methods which allow correction of the gene defect that has been diagnosed in a child or embryo. Particularly if it is done in the case of embryo, then the child which is going to get birth or get life will not be having that type of hereditary disease. In the sense, for example, take a couple who are having genetic disease. And what we are supposed to do in the sense, we have to collect ovum and sperm of those that couple, and the gene therapy has to be done. That means finding out the defective gene, taking out that gene and replacing with the healthy gene, and then it can be left out for fertilization. And then naturally the embryo that is the resultant of that particular zygote will be of healthy and not having any of hereditary disease. Okay, so that type of technology is seen in gene therapy. Okay, so tomorrow we'll continue.